really feel qualified to be a mentor here, but I'm just hoping that what I will tell you in the next half an hour or 20 minutes or so, you will find something in there that you can make use of. So here goes. And before I start, I would like to really thank the organizers for putting together this beautiful template and pointers on all the slides and what you should say. I found it really useful. So that's all. Okay, so here's my background. My first degree was a BTech in industrial biotechnology. This was really a very unique program back then at Anna University in Chennai, which was in an engineering school. So we did engineering for the first couple of years and then did life science subjects, and it was a very packed and exhaustive curriculum. But I really enjoyed it. We had great instructors and they were all very research minded. And really, this program, 70 to 80% of the people went on to do their PhDs. So this was like a path that was not even questioned by most people. They just went on to get their PhD somewhere, and that's what I did. And you'll see that this has been a trend in my life, just to go with the flow kind of thing. So I went to Cornell to do my PhD, and after rotating through multiple labs, I joined the lab of Professor John Bliss. He is a person who's known for working on forced polymerases, and he used the Drosophila model system in the heat shock promoter to look at gene expression regulation, and he was a big personality in the field, and he would take students from completely different backgrounds and encourage them to do whatever they wanted. He was really had the money to do it and encouraged people to do whatever they wanted. But even though he was known for transcription, he had a small subgroup that worked on aptamers. So he thought that aptamers, which are small nucleic acid antibodies, you can call them that, you select them by an in vitro selection process, but then they bind with high affinity to the target protein. So you can use these to tease apart protein-protein interactions, and he thought this was the way to go. This is how people study protein interactions and so on. That was his idea, but he convinced a bunch of us to work on that, and I learned a lot during my PhD doing that. After this, towards the end of my PhD, I had indicated to John that I really enjoyed working on research questions. But I didn't really want to have a lab of my own, so I was considering career in industry. But he was like, no, you should not do that. Do at least a postdoc and then decide to change your mind and so on. So at this transition, I also had, this was the time that I also had a child. So I took a year off, but in that time I interviewed in various labs and ended up in the lab of Danish Muazzar, who's another gene expression person, but he works on silencing in the fission yeast model. So he was studying how heterochromatin forms and what are the various pathways in RNA-mediated silencing, how is it established at heterochromatin and so on. And being a young mother, I was really looking for labs in Boston which didn't require me to spend 24 hours in the lab, which was a whole bunch of labs I interviewed at seemed like that. So I chose Danish's lab because he was a very, very supportive mentor. But again, a very big guy, no problems with funding, extremely intelligent, and gave everybody a free rein to do whatever they wanted. So these were the two PIs that were my advisors for the 10 years I spent training. And so after looking at the two of them, my intention of leaving academia became even more solid. I was like, there's no way I can do what these guys are doing. And it was also coupled to my move to India. So we had decided, my husband and I had decided that we would come back after I finished my postdoc, or after this H1 completed 6 years, because we said there has to be an end point in sight, otherwise we would never do it. So we made the decision to move back to India, and I was definite that I'm not going to academia, certain. So a bunch of things influenced this decision. So like I said, it was a daunting possibility to have to manage, your own lab and get funding and manage students, and it just seemed very complex. I really, really did enjoy research though. So I thought R&D and industry would be like the perfect fit. And a lot of other things, young child in tow, and I was by nature quite anxious and don't like to network. Like in India, I've noticed that people generally just pick up the phone and call people and talk to them. You have to call granting agencies and be like, where's my money? These are not things that I would ever be able to do, and I knew that. And I still don't to this day, which is probably like a big part of, that's just how I work. So with all this in mind, I did apply to a lot of industries. Hyderabad has a lot of pharma companies, and I did get offers from many of them as well. But I found out soon enough that the job didn't involve any research. It was just people management, some paper writing, and not really exciting. So I also knew that I couldn't survive in a place that was not intellectually rewarding. So I had to find something else. So I spent almost a year talking to people from various fields. I considered moving to a journal, or there was somebody in a business school that I knew of who was doing something unique, who also had a degree in the life sciences, a PhD in life sciences. So through her I met somebody who was at Dr. Reddy's Institute of Life Sciences, and this place, I had never heard of this place, and as I'm sure most of you have not even to this day. So I met someone from there. He invited me to come give a talk and said, you should come here, it's a small place, but you'll get the freedom to do what you want, and blah blah blah. So I joined Dr. Reddy's Institute of Life Sciences, and that's where I've been since then. There was a short interlude where I was also part of a startup, but I don't think I'll have time to go into that right now, so I can talk to people later if you're interested. So Dr. Reddy's Institute of Life Sciences is not Dr. Reddy's, the pharma company. Let me make that very clear, because I think a number of people have already asked me that. It is a public-private partnership kind of setup. So Dr. Reddy's, or Dr. Reddy himself, was interested in setting up a company and a research institute which would cater to the needs of industry. So it's a place where he thought a lot of the early R&D could be done, because this cannot be accomplished in an industry setup where they have some rules and regulations and GMP and all of that. So he thought that this would be a place where multiple industries would contribute, come there to get their problems addressed, and a bunch of scientists who are interested in doing that kind of work could be recruited and could cater to these needs. It didn't quite pan out that way. So Dr. Reddy's continues to be the only industry that is invested, although recently it's been changing a little bit, thankfully. But what I was told that was the expectation from the PI would be that you would still get 50% or so of your time to do whatever you wanted, as long as you could find the grant funding to support the work that you wanted to do. But as and when the need arose, there may be, you may be required to do either industry-sponsored projects, or the institute may decide that this is a focus area for the institute and may want you to work on some projects in that space, and you should be willing to do it. I thought this was a good win-win situation for me. So I said yes, and we started here. But as with any other institute, I think there are both pros and cons to being in a place like this. It is a very small and very unique setup, which means there's not much red tape. We are supported by a very small but efficient admin team, and we can reach the HR or the finance, which is all essentially just one person, and get things done very fast, which is a big pro. But it is also a con because there are no set processes. It's not like you can negotiate a startup package, or you just have to do what they tell you to do in terms of, like for example in my case, I got a very generous lab space with equipment that was left behind by the PI who was there before, which was great for me. I didn't have to build anything up like many of the other PIs. But I also didn't get any startup package. So I had to wait for my funds to roll in before I could actually start doing anything on my own. So that's where I started. So in the initial phase, back to being an academic, I had never really thought about what I wanted to work on. So I relied on what I had done until then. I did always have an interest in looking at non-coding RNA and how it regulates protein interactions and how it influences gene regulation. This was something I had been interested in, and I had worked on both in my PhD and postdoc, and I still found that interesting. And since you have to have a demonstrable or demonstrable track record to apply for grants in the beginning, I used that to write the first couple of DST-DBT grants, which thankfully got funded. And while I was waiting for funding to start was when I interacted quite extensively with my colleagues at the institute. Everybody was doing something different. So there was a nanochemist, there was a vet turned pharmacologist, there was somebody who was a molecular biologist, cell biologist, a lot of metabolism and metabolic dysfunction people. So we used to have all these conversations, and through one of these, because I had some experience with working on aptamers, I got roped into this startup thing. So YIM had just started in 2012, and 2-3 people got together to set up this nanoparticle mediated drug delivery platform. And because aptamers could be used for targeting, they asked me if I would be willing to join the team and contribute to these projects. So I did that. And while I was discussing with these people, one of the same people was also using zebrafish as a model to test these delivery platforms. And so since there was a nice zebrafish facility set up by Drills, thanks to Kiranam and Anil and other people who were there before me, I got introduced to this model system. And so through these discussions is when the whole idea evolved that why not use the fish model to create, there were people who were doing chemically induced models and then were interested in neurodegenerative disorders. Being associated with a pharma company, you're always expected to somehow pitch into screening programs or something like that. But there was no really genetic model that was being worked on by people there at that time, and I was interested in it, so we got together and said, okay, we'll use DNA zymes, which are kind of like RNAi, but RNAi is not really popular in zebrafish. So use DNA zymes to create transient knockdown models and have a platform where you could screen for phenotypes. So the first disease that we started with, based on an interest in autism for the PI that I was working with, was Fragile X syndrome. So we developed a platform to use DNA zymes to knock down FMR1, which is the gene implicated in Fragile X syndrome. And it worked really well, and we got some behavioral phenotypes, and he was able to do some screening. And we sold this as something that you could use for other rare diseases as well. So Fragile X happens to be a rare disease. So we said, you can expand this platform, not just use DNA zymes, but use CRISPR or any other available technologies to create models and make them available for the community to develop therapies, to identify biology and pathways and so on. I found this interesting because it also allowed me to do biology with these models. So rare genetic disorders basically allow you to study each gene in an organism model with the direct human context that is already available, because it's disease and it's translation even before you start, right? So it worked out. There was, of course, a learning curve because I had never worked with fish before, being a yeast person and then before that just an in vitro RNA biochemist. This was a hard transition, but that's where the people come in. I think everybody has told you that peers, mentors, your students sometimes, people who've been in the facility for a long time, everybody is a teacher, and I generously leaned on everyone to get as much input as I could and learned on the go, and that worked out for me. The one big challenge I always had was this whole concept of applied versus basic, which I see many of you are still wondering about. I don't think that ever gets actually clarified, but the take that I have on it is we find the basic in the applied as and when it happens. So, for example, rare disease biology or working on rare genetic disorders, creating models is considered applied research, right? But I work on FMRP, which is an RNA binding protein, and I'm interested in studying how this RNA binding protein, how does it regulate translation? Is it only doing it in a translation? Is its function limited to the RNAs that it binds, and by regulating translation is the only way it has impact, or does it do other things? So these are questions that can be considered basic, but it's in the context of an applied research. So as long as you can sell it in that way, I don't think you have to really stop doing what excites and motivates you, and that's how I look at it. So the big learning that I have for grant strategies from all of this is, like somebody was saying, write your grants well. I guess that's what was told to me. We have this habit of just circulating the grants among our own department people, and everybody reviews it and gives you comments. And all of these people who were there before me had multiple grants, so that was really useful. The other thing that they told me was apply to as many agencies as possible, as many times as possible, until you get the first few grants. Once you have that, then you can be a little bit more selective about, okay, I'll pursue this one and I'll not pursue this one. The other thing is also that even inside these agencies, there are multiple committees. So there is, in DBT at least, I don't know if it still exists, but there used to be a basic science panel, a human genetics panel, a genome engineering technologies panel. So if you write one to each of them, then at least it's not the same review committee that's seeing you and rejecting your grant again and again. So you can get more success out of that. So these are some things, and I also got on to many co-PI grants because some other PIs require a fish model for something. So that's an easy way to get a small pot of money to keep you going when you're waiting for yours to come in. So then a couple of things happened. I think 3 main things happened, which kind of were transition points in my career. The first was that I was appointed as this research coordinator for the Rare Disease Initiative at DRIS. So based on the first few successes that we had working on small rare disease projects, a lot of clinicians were interested in coming to us and saying, can you create a model for this gene? We are finding this in our patients. Can you do this for us? Can you do this for us? So the institute, the board, and the research council members also advised us to kind of make this a focus area for the institute. It works well with the theme of the institute being having some translational aspect to your research. And it also gives us an opportunity to work towards therapies, maybe not immediately, but eventually in a 10-20 year timeline. The main point that I thought was very useful was that there was truly an unmet need for models for rare disease biology in India, because not everybody can afford rodent models or a rodent facility. So using zebrafish was a good thing in that way, and we were able to create multiple models. We have about 15 in the facility in the past 5 or 6 years that we've created, and I think that has been a very useful contribution. And of course, this was by the way, not the motivation for doing this, but it did check a lot of the boxes for the buzzwords. It's translational, it's rare disease, and this was just before rare diseases actually became a buzzword, which happened I think in 2018-19, so that was useful. And it was also a good match for my scientific style. I got to work on a lot of different things and didn't have to dedicate my life to that one area, because I truly, honestly don't think I have that clear in my mind even today. And it was particularly well suited for multi-institutional grants. So this was the second good thing that happened, is that we were asked by KMC Mahe to be on a grant with them, a clinical research grant, where we would do the functional validation for some of the genes that they identify in their patients. And getting on a grant like this is really a good boost for any program that is at its infancy. So then COVID happened. So this kind of put the brakes on everything that was going on. But there were a few good things that came out of this. One was, of course, being on the CRC, because this grant was entirely written during COVID, and we got to know the results after the second phase, I think. The other thing we did was a lot of people approached us for working on small projects, both clinical and from industry. And I won't mention all of them, but the last one, where there was a company who was interested in setting up a neutralization assay, a pseudovirus neutralization assay that one of my colleagues did for them. This was a need at that stage because there were only a few companies and they were located in other parts of the world, in Canada or something like that, and access was difficult. They couldn't send all their sera out and get it screened. So having somebody local was really useful, and we agreed to do that. And what that has done since then is that they've come back to us with multiple requests on completely unrelated projects. So I think showing that we are willing to work with them and do whatever they needed at that time, one, introduced us to them and demonstrated our capabilities of being able to work with them on a timeline, do what they want. But also now they're more willing to listen to our ideas and incorporate them into what they're doing. This of course means that you have to deviate from whatever it is that your whole focus area is. But I think it's very rewarding because, at least for me, it's something that has more immediate societal impact and is a pot of money that's not the grant stuff. I have 10 minutes, right? 5?
10 minutes?
Okay. Yeah. Okay, so back to, I will go through these slides very quickly. So when I say rare disease research, what we essentially mean and how I divide this up in my mind is, I have 3 buckets in which I bin these diseases and models that we create. I am interested in the science of a few of these. So for example, the Fragile X model and the PISD model. These are things that I have PhD students working on, which means I'm interested in working on them for 5 or 7 years or 10 years or maybe forever after, if I can continue to find support. So even though these came out of the CRC, I've written independent grants for these specific projects and I continue to work on the basic mechanism aspect of that. But in exchange for being part of the CRC, I also have to do functional studies for the clinicians because that's what they want. So we always have a couple of projects at least where we do short-term studies where we essentially help them prove that this is the variant or gene that is causing the phenotype that they are seeing in their patients. And for the institute to keep giving me support and in kind and cash and whatever ways I want, I have to do some programs that they are interested in. One of the ones that they are interested in is therapy. So that for that we have to create some models, and that is for the therapy development aspect of it. So I'll skip these slides. I'll just talk about 2 specific examples of this therapy thing. This because we developed a model for Duchenne muscular dystrophy or DMD in zebrafish. Zebrafish is a well accepted model organism to study this disease, and there are a number of people who've made models. But because we had this, and we were able to set up an assay to monitor the locomotion defect that these larvae have because of the muscle integrity defect that they have, we were able to use this assay to collaborate with a company who wanted to screen their compounds in our model. And we were able to give them data, at least for one of their compounds, to show that the molecule did improve locomotion. The mechanism and the reason for how it worked and all of that is their work. So here we're basically just providing a service, but it gives us another pot of money. So using this, they were able to seek more funding and they're moving their molecule forward, which is heartening to know. And we've also created another model. This is for an inborn error of metabolism called methylmalonic acidemia. I'm just showing this to show you that we use our models in different ways in addition to doing the basic science that I'm sure all of you have your own areas to work on for that. So here we set up an mRNA platform back during COVID times when people were interested in making mRNAs for various variants of spike and so on. We repurposed that to create an mRNA therapy for this disease. So this is a mitochondrial protein, so the standard ERT will not work. But we developed an mRNA, and in the case of MMA, what happens is this metabolite called methylmalonic acid accumulates and causes liver and kidney problems. So the zebrafish model also shows MMA accumulation. And then when you dose it with a single dose of the mRNA that we prepared with an injection, you see a reduction in the metabolite. And so this we've been able to write an ICMR grant with this and get funding to take it to rodents and also use this to develop AAV-based therapies as well. So with that, I'll spend the last 5 minutes just going over some challenges and solutions. I think a lot of the other mentors have covered many of these things. So maybe I'll just focus on 2 things. So being a private institute, finding funding for facilities has always been hard. I know recently there are some possibilities for even private institutes to access this funding, but what we did was, because of the rare disease work and how it applies to, how it appeals to philanthropic sources of funding, we were able to seek CSR funding to buy a lot of equipment and set up a new facility called the Center for Rare Disease Models, which is what you're seeing here. And that has been really useful for us in terms of getting more, husbandry racks and high-end equipment and all of that which we can't get from grants. And in terms of hiring and building a research group, because the program that I run is not really, one question and the lab works on this and has been working on it for several years and so on. It's also hard for me to find students and train them in all the different things that we do. So each gene that I work on that comes from a physician or comes from someone else requires a different set of skill sets. And not everything can be converted to a PhD project. I don't feel like it's fair to ask people to do things that even I am not trained on or cannot help them on. So I don't take very many PhD students. But I've had great luck with undergraduates and master students. I've had about 20 to 25 of them go through the lab. And as I was telling someone else just before lunch, I think convincing people or telling them that you should do a PhD is one thing. But keeping people who are not ideally suited for a PhD away from PhD is also an equally important contribution, I feel, because not everybody is made to do that and you don't really need it. Especially in India, if you see a lot of master students come to you because they don't have anything else to do and the PhD pays you a stipend, so they treat it as a job. But taking on students like that is more mental headache for the PI, and you're actually doing them a disservice in the long run because now they're stuck with a degree that they still don't know how to use. I'm sorry to be very brutal about this, but that is the truth. So I think I've had a lot of great master students who are convinced to apply abroad and go because they'll get a better training from a lab that is a more traditional academic lab than mine. But I've also had some of them stay behind, and especially my current PhD student, one of them is someone who probably wouldn't make it past the cut because of her inability to communicate or inability to express herself or something like that, but she is PhD material. So for someone like that, I feel like I should be able to put in that extra effort and help them out, so I do that. The other thing is also micromanaging versus not, and a lot of the people that I see in the lab don't really need help, and in those cases, I leave them be. And I think that luxury comes only with time, because in the initial years, the granting agency requires you to show progress. And if you let the PhD students just do what they want and they don't do what you need, you will not get the next grant. So you have to kind of be a micromanager in the beginning, but then later on, when you have the luxury of multiple grants and using money from one for the other, I guess you can let them have a free reign if they deserve it. Okay, so I'll just quickly finish up. I also enjoy, like I said, teaching, mentoring and all of that quite a bit, and I've had high school students come through the lab and do a lot of very crazy fun projects, including my son, who I don't know if you can see over here. He worked on the DMD model as well and was in 8th grade. And I've also convinced non-science people to kind of try and make them more excited about the science that we do. And these are 2 illustrations that were created by 2 art students, which now adorn the walls of our new center. And I think they really did a great job capturing the essence of the Center for Rare Disease Models. And what I hope to do in the future is to continue. I finally feel like I've found my balance, and I hope to continue to be able to do this and do it well. I think that's what the future should be for me. And I do hope to continue to do this industry-sponsored project. I now have a couple of people who are dedicated to only doing this. So hopefully we'll go on. I think that's it. I'm happy to take questions later if anybody has.